Bismillahirrahmanirrahim. Dear students, today's topic is the description about the role played by the biotechnology in disease diagnosis, and particularly we will discuss DNA, RNA probes, and monoclonal antibodies because this is the part of our syllabus in MDCAD test. So let's start with the proceeding. So role of biotechnology in disease diagnosis and particularly DNA, RNA probes and monoclonal antibodies. So as we know, biotechnologies plays a vital role, pivotal role in disease diagnosis by providing highly sensitive, specific and rapid method for detecting pathogens, genetic abnormalities and disease markers which may be present in a healthy person and for the detection in future onset of the disease. So two key biotechnological tools, they are under discussion. For example, DNA, RNA probes and monoclonal antibodies. So if we talk about DNA, RNA probes, so DNA and RNA probes are short, single-stranded nucleic acid sequences, and they are being designed to bind to complementary target sequence in the patient sample. So they are labeled with detectable markers, most of the time fluorescent dyes, which are inert, and sometimes radioactive isotopes, which may be necessary in some cases, and the purpose is to visualize the outcome. So application of DNA RNA probes in disease diagnosis. So number one is detection of infectious agents. So the purpose is to identify genetic material. It may be DNA, maybe RNA of pathogen like viruses. For example, in case of COVID-19 via real-time PCR, maybe bacteria, and particularly about TB bacteria and some other parasites, we may be multicellular in nature. So enable early and accurate diagnosis even at low pathogen concentrations. So this is very important because this may be early detection, rapid detection, and the detection at what we say out of the total population context. Uh, then comes the diagnosis of genetic disorders. So detect mutations or deletion in genes associated with disease like cystic fibrosis, sickle cell anemia, Huntington disease, and used in parental screening and genetic counseling. So, for example, in case of this leukemia and thalassemia. So cancer diagnosis, identify oncogenes, tumor suppressor gene mutations. For example, if a person is having a suppression of those genes whose function is to suppress the tumors, so mutation may cause the abnormal function of them. Or chromosomal abnormalities to classify cancers and guide treatment of the patient and helping doctors and physicians to cope up with the problem. Then the pharmacogenomics. So this is important to analyze the genetic variations to predict individual response to drugs, enabling personalized medicine. This is important because generalized medicine may have some side effects, and if we have personalized medicine, that may be safer, cheaper, and targeted. So what is the principle behind this DNA RNA probing? So the purpose is to help the hybridization, mean binding to complementary sequences to confirm the presence of specific genetic material. The sole purpose is to hybridize, and rest of the functions, rest of the what we say benefits we get out of it depends upon this hybridization of the DNA of our labeled administration and the patient's DNA. So then comes the monoclonal antibodies, abbreviated as MABs. So monoclonal antibodies are identical antibodies produced by a single clone of cells targeting a specific antigen with high precision. So how to make it? It's very simple. We first of all, we identify a B cell producing some specific type of antibodies. Then we hybridize it with hybridoma, and to make the cell which is having the ability to divide and redivide and having the ability to produce monoclonal antibodies. I would say technique very useful. And its application in disease diagnosis, for example, detection of antigen pathogens. For example, we may perform ELISA test, and the purpose is to quantify the antigen, maybe viral proteins or antibodies in the sample. So ELISA is a little bit cheaper and rapid method as compared to the real-time PCR and rest of the samples. So then the rapid testing used in pregnancy tests, COVID-19 antigen test, and other point of care diagnosis. Uh, uh, this is very simple. For example, we may be having sticks, and those sticks may be having some sort of antibodies attached over it, and by applying simple sample material, we get the result. So then comes the immunohistochemistry, visualize the disease markers in tissues, for example, cancer biomarkers, and then the serology in order to detect the antibodies in blood to diagnose infection, for example, HIV, hepatitis, or autoimmune diseases. So blood typing and tissue matching ensure compatibility for transfusion or organ transplant by identifying blood samples, blood group antigens, or HLA, mean the human leukocyte antigen markers. So then we apply monoclonal antibodies through flow cytometry, and this is very effective instrument in which we actually pass through the sample, and the laser beam will detect the specific type of antigen in order to tell us about the presence of leukemia cells or something else. And it may give us the idea about the number of those cells and their shape and the markers. So this is very, what we say, rapid, easy, but quite expensive. So in vivo imaging, locate tumors or infectious agents using monoclonal antibodies tagged with imaging agents. We may use them as PET scans and through MRI machines. So the principle behind it, that monoclonal antibodies exploit antigen antibody binding for specific detection purpose. So there is a table which give us the idea about the biotechnology tool and its key application. For example, DNA, RNA probes detect genetic mutations, infectious pathogens, and cancer-related genetic changes. And monoclonal antibodies, they will give us the idea, and we use them as to identify the specific antigens. Antibodies are cells in disease like cancer, infections, and autoimmune disorders. So these tools revolutionize the disease diagnosis by enabling early, accurate, and targeted detections, ultimately improving patients' outcome and the cost-effective method. These are the cost-effective methods if we consider them on what we say diagnostic purposes. So in order to re-summarize and what we say, in order to provide the revision, so role of biotechnology in disease diagnosis, biotechnology enhance disease diagnosis through precision, speed, and sensitivity. These are very important points: precision, speed, and sensitivity. So key tools like DNA, RNA probes, and monoclonal antibodies enable early detection, accurate profiling, and personalized treatment strategies. So if we revise DNA, RNA probes, so definition: short, single-stranded nucleic acid sequences labeled with markers, maybe fluorescence, to bind complementary target DNA, RNA in the given sample. So key application in infectious disease detection: detect viral RNA For example, in case of COVID, in case of SARS disease, or bacterial DNA, Mycobacterium tuberculosis, or identify the drug resistant strains, MRSA, to guide therapy. So genetic disorders screen for mutations. For example, many genes which may cause the breast cancer or something else. So we can screen those mutations if that person is having or not. So parental diagnosis, for example, the Down syndrome via FISH method, and cancer profiling tumor specific mutations for the targeted therapy. The advancement in this case, CRISPR-based probes, so far portable low-cost diagnosis and microarray technology to analyze thousands of genes simultaneously. So advantages: high specific complementary base pairing, can detect low abundance targets, for example, having HIV virus, but the outcome of disease was not at that time. So monoclonal antibodies definition: uniform antibodies targeting a single epitope. The epitope is the protein or maybe some other structure present on the pathogen surface, which can enhance and produce the antibody response, and that is epitope, produced by hybridoma technology or recombinant methods. So key application: diagnostic test, ELISA, in order to perform the quantity biomarker by using quantity biomarkers, and it can give us the idea about the presence of certain diseases as well. So latent flow assays, rapid tests, for example, malarial test or some other tests. Histocompatibility, visualize the tumor markers, infectious diseases, detect viral antigens, autoimmune diseases, and then the blood testing or tissue typing, ABO or RH blood grouping, HLA matching for transplants. And then the emerging uses of this method is biosensors for real-time monitoring, glucose sensors for diabetes, multiplex assays to detect the multiple pathogens simultaneously. So advantages are exceptionally specific. For example, in case of lock and key antigen binding, so it will give us the idea of those two interacting agents. So versatility using in imaging, therapy, and diagnosis. So monoclonal antibodies are very effective. So comparison table, if we look for the feature, so they target DNA, RNA probes nucleic acid. Their target is nucleic acid. And monoclonal antibodies target is proteins and antigens. Sometimes they are also proteins. The technique principle in case of DNA, RNA hybridization, the probes, the hybridization. And in case of monoclonal antibodies, antigen antibody binding. So common uses: DNA, RNA probes, genetic disorders, pathogen cancers, monoclonal antibodies, infectious diseases, cancer, autoimmune responses, and example techniques: PCR, FISH, microassays, and monoclonal antibodies, ELISA, and flow cytometry. Future directions: integration with AI. This is important. Machine learning to interpret complex probe antibody data. So it will give us the idea and detection will be more easy and precise. Point-to-care devices, portable devices, for example, CRISPR technology will be used in case of certain kits for in order to get the idea in the field. For example, no need to go to diagnostic labs, just collection of samples and get the result, the diagnosis. Then the Theranostics, Theranostics combined diagnosis and therapy. For example, monoclonal antibodies linked to drug for cancer. So biotechnology continue to transform diagnostics, offering faster, cheaper, and more accurate solutions to improve global healthcare outcomes. So that is all about the DNA, RNA probes, and monoclonal antibodies in case of diagnostic purposes. So that is all about this lesson. So we may come together for next lesson.